Webinar brought to you by Syntica. My name is Martin Hess, and I will be your host for today's event. Our webinar today is entitled How to Use MycoFog to Reduce and Prevent Contamination in Tissue and Cell Culture Laboratories. Today's session will be presented by the dynamic duo of Randy Engler, the CEO of MycoFog, and Samir Patel, the CCO of MycoFog, both of them co-founders of the company. A brief introduction to both. Randy is a biotechnology business executive with years of global business development, product development, and sales experience in all aspects of the biotech industry. He has academic training in microbiology and immunology, and has led commercialization efforts and teams introducing new products and technologies to new markets for companies as diverse as Genzyme Diagnostics to Thermo Fisher. Samir has over 4 decades of international experience in commercial leadership within the life sciences and healthcare industry. As managing director at BioTrace, he established market leadership in hygiene testing. And as global sales director at the Baker Company, he expanded the global adoption of gas-controlled hypoxia and anoxic workstations in microbiology and cell biology labs. Now a co-partner at MycoFog, Samir is focused on advancing innovative biodecontamination techniques for cell culture incubators and glove boxes. So I'd like to mention a few quick housekeeping items. We anticipate today's presentation to last roughly 30 minutes, the webinar from start to finish. It will include a question and answer session at the end. I would encourage everyone to please submit your questions through the Q&A dialog box that you see on your toolbar on the screen. You can submit questions at any time through the presentation. Your microphones are muted, but we will address any questions you submit at the end of the webinar. We will also create a written transcript of the Q&A session, and we'll make this available for distribution over the coming days. Also to mention is that we are recording today's session, and we'll provide a link in a follow-up email so you can access the content again at your leisure or share with your colleagues. Okay, before we begin, we have a couple of quick poll questions that we'd like to ask you. And this will allow us, I guess, to better understand how you currently go about decontaminating your cell culture incubators and glove boxes. Okay, so we're launching a poll. You should see 2 questions. The first question is, how often do you clean and decontaminate your incubators? Please select one of the options listed. So they are when there is a contamination, that's option 1, regularly, either monthly or quarterly, once per year, and then there is another option, maybe you don't decontaminate, you should, but whatever that other option is, feel free to enter that in the chat window. And then question 2 is, what method do you currently use for decontamination? The options are built-in, so high temperature cycle or other incubator-based method, wiping with alcohol, wiping with other decontaminating compounds is the third option, and then there's, again, another other option there. I'll give you a minute to just enter your responses to those questions, and then as soon as we're done, we will get our session started. Okay, thank you again. And again, just a brief reminder before we get started, feel free to ask questions through the Q&A dialog box at any time during our session. You're here today because you obviously care about preventing contamination in your incubators and workstations and learning how best to do this. And we'll be highlighting a simple and effective method to do so today. So with that in mind, I am going to pass controls over to Randy to begin his presentation. So Randy, whenever you're ready, feel free to unmute yourself and begin your presentation. Thank you.
Thanks for that really kind introduction, Martin. I appreciate your commentary. And I want to thank all of our audience members for taking the time to attend this webinar to learn more about biodecontamination of your laboratory instruments, your incubators, and closed workstations. We'll start today with a brief introduction, and we'll talk about the types and sources of biocontamination, talk about various different types of disinfection methods and approaches to biodecontamination. And we'll talk about how MycoFog can help you with your contamination control in your laboratory in a easy, efficacious, and economical manner. And then we'll get into some details about the type of equipment that we focused on in developing our products. The first thing that I want to talk about is that contamination of cell cultures has been around pretty much since cell culture started. I have a quote here by Don Armstrong, a prominent researcher who was doing cell culture back in the early 70s. And he said, as an introduction to his book, contamination of cell cultures by bacteria and fungi almost invariably results in ill humor in the laboratory, lasting from hours to days. One or many discarded cell cultures, and accompanying these two major reactions are the following: soul searching, culturing of all media components, including the thioglycolate broth, scrubbing of the laboratory, tightening of techniques, and no further contamination temporarily because of the activities mentioned in items C and D. So you see, people have been struggling since the early days in cell culture in using their in trying to get a good decontamination routine. Today, we're going to talk about the types of what we're talking about when we talk about biodecontamination. We talk about all sorts of microbiological organisms, bacteria, mycoplasma, yeast, and fungi, and viruses, and of course, all sorts of different types of viruses. What we don't discuss and address in our products is protein contaminations, particulates, or chemical contamination. We're strictly discussing today biological contaminations. Now, how do laboratory incubators get contaminated? Well, their contaminations can be introduced in a number of different ways. There's airborne particles that are constantly in the air, and some of those dust particles may be carrying organisms or spores of organisms. Of course, the user interaction is a significant source of potential contamination. Handling samples, you must use aseptic technique, and a lot of contaminations are caused by a breakdown in aseptic technique. Excuse me. Moisture and condensation, of course, which is the environment that we, well, not condensation, but moisture is the environment that we attempt to achieve in the incubators. But these will provide the ideal environment for organisms to colonize and grow. Cross-contamination. So if you introduce a contaminated culture into an incubator where there are is no contamination, there's a reasonable probability that contamination can spread to the other cultures in that same incubator. And as we just as the last item here is the thing that we're trying to avoid. Even with cleaning, microbes can linger on surfaces, and we'll today talk about very specifically how to address that particular item. And then we're going to talk next. We're going to talk a little bit about corrective and preventive cleaning protocols. Uh, kind of two different approaches that people take to maintaining their incubators. Before we get into that, I want to talk a little bit about the very earliest stages of things that you can do to minimize biodecontamination in your cell cultures, and that is your lab layout. I'm not going to go into many details about this particular layout, but the point is that you want to make sure that your incubator are not opening in a high traffic area. So you want to make sure that the incubators are in a corner where there's not, or in a part of the laboratory where there's not a lot of traffic, because more people walking by creates eddies in the air and stirs up potential biodecontaminants. So now I realize that most of us don't have the luxury of designing our lab from scratch. And so we have to think about where we put our equipment in the lab in order to minimize the potential, the risk of contamination. There's a number of sources of contamination that we mentioned air eddies, which we can see in the cartoon in the middle of this page. Every time you open the incubator door, you're pulling in or creating eddies that have potential to introduce contamination from the air inside the laboratory. Also, all of your media components that we kind of indicate here on the left, serums, bovine serum, any supplements, everything needs to be, of course, contamination free. And this means you have to have a good incoming quality control protocol or have reliable vendors who have their own protocol for minimizing the possibility of contamination in their in the media components that they supply to you. The other thing is maintaining aseptic technique in all of your operations is really important. And in this cartoon on the right, we can see, we've got a biohazard hood work area that is pretty cramped, filled with stuff, and that's going to affect the airflow within the biosafety cabinet and maybe reduce its effectiveness in doing its part of the contamination control in your laboratory. So you have to be mindful about the way you set up and store items in your biosafety cabinet, as well as on your lab bench and in your incubator, that can create kind of a nightmare cycle of reintroducing contamination if one of those components isn't taken care of properly. As I mentioned earlier, there's a couple of different approaches depending on the decisions that you make in your laboratory, and they're not hugely different. But in doing corrective action, of course, somebody has to do some observation and potentially testing to find out, is there a contamination? If that answer is yes, then you need to go through some of these procedures, removing all the contents, cleaning everything, and we usually recommend cleaning with soap and water, as we saw in the in the user survey earlier in the presentation, some people are using alcohol to wipe down their surfaces. We think we have a better way. We have a biodecontamination system that actually vaporizes hydrogen peroxide into your chamber, and it actually penetrates all areas. So even residual organisms can be killed if they remain in the chamber. The water pans have to be replaced with clean, sterile water. And that should be done on a regular basis. And you can then test the efficacy of your cleaning and decontamination using biological indicators, which are we can get into that a little bit later in the presentation, but they're actual spores that are resistant to killing, so that you can determine, but they are killed by the hydrogen peroxide process, and so that you can determine this success of your own decontamination processes. The next kind of broad category of cleaning is preventative cleaning. And here you, instead of waiting for a contamination to happen before you clean your incubator, you do this on a regular method in order to prevent those potential contaminations. And this means routinely wiping it down. As I mentioned, we like a mild soap and water for cleaning, and then do a regularly scheduled biodecontamination using MycoFog. Of course, we're a little bit biased here. We like that methodology. It's really more efficacious than wiping, and that should be done on a monthly or quarterly basis. Make sure you replace the water in the water pans regularly, weekly, and also run a biological indicator check. Now, as you can see, the difference between preventative and corrective is that there are fewer steps in the preventative. And so that's one of the reasons that we're a big fan of doing it regularly before you run into a contamination problem. And with that, I'll turn it over to my colleague, Samir. I'm going to stop sharing. Samir, it's yours.
Thank you, Randy. Good morning, good afternoon, good evening, everyone. Please select according to the geography you're in. And also, again, thank you to Syntica for organizing such good webinars. This is the second or third, maybe the second one we've participated with them. Excellent. So let me introduce MycoFog. This is the answer to your incubator and biodecontamination needs. And the photo here shows the system, the complete system. On the right-hand side, you can see an instrument. We call it a fogger. On the left-hand side, you can see a package of 4 pods. Each of these pods has hydrogen peroxide in them, and each pod will do 1 decontamination cycle. We'll get into that in a little bit. But to give you a relative feel for the size of this is 10 centimeters high, 10 centimeters in diameter, and it weighs around 400 grams. So you can pick it up in one hand. We'll show you that in a second in a video. And also worth pointing out that the reagents all come pre-dispensed, ready to use, and they only need room temperature storage. Another nice feature of this instrument, the fogger, is that it's battery driven. So you would charge it up with your USB cable, which is provided in the little kit, and you charge it up the day before, and it will do up to 6 cycles. But we would ask you that you keep it charged every day. So now let me just show you on the next slide here. We have a small video of the MycoFog being used. You'll be able to see it and gauge its size and feel a lot better if I start this. So it can easily be opened by twisting the top off. There's a reagent container in the center into which you pour the hydrogen peroxide. place it into the incubator. We'll come on to that. And then watch as the fogger generates its plume. You can close the incubator door now and walk away. And then this plume of hydrogen peroxide will fill up the target incubator volume. Very simple, very easy to set up. So what's happening and what do we have to do in the background? This is a simple process, it seems like in the video, but you do have to take care that you empty the incubator in advance of all the cell cultures. They have to be put into another incubator. You can set the temperature of your incubator or workstation to 37 or 45C. We have two different protocols. Turn off any gas control to that incubator or workstation, turn off any humidity, and then you're ready to put the MycoFog inside. Once you place the MycoFog inside and you switch it on, as you've seen, you close the door and you walk away. You're going to walk away for either 4 hours if you're using a 37C protocol, or 3 hours if you're using a 45C protocol. And during this time, hydrogen peroxide is generated from the fogger into an airborne form. It's going to reach all parts of the incubator, kill all the biological material that Randy mentioned earlier, and then it will also break down and dissipate, all during the same time. You don't have to do any more interactions with the MycoFog. So then that just brings a little quick chemistry reminder to you. Why hydrogen peroxide? Well, it's a very strong oxidizing agent. It loves to strip electrons from things, therefore it breaks down cell walls and bacteria and viruses, walls, spores, and so on very well and very efficiently, especially in a broad temperature zone of 28 to 50. But I've told you already, we have optimized our protocols at 37 and 45C. The ultimate residues left behind are going to be water and oxygen, and there should be no H2O2 at the end of a cycle. I'll come on to that again in a second. So let's just see what kind of target instruments we have our customers using right now. Typically CO2 or trigas incubators. That's our biggest target zone. That's what we've been talking about today. If we open this door, you'll see inside the Heracell 150. There's the fan. There's how the MycoFog looks inside. Just before we close the door, you can still see the plume working there. And interesting to show you this point, because the shells have all been placed to the bottom underneath the MycoFog. We'd ask you to put them in the lower half of the incubator to allow the plume to have free access above itself, above the generator. Just showing 2 shelves here, but sometimes you get them with 4, sometimes with 6, but you can put them in the slots that are underneath and maybe even raise this one up. Here's a climate control chamber. He hasn't moved the shelf yet to below, but just to show you, a larger climate control chamber also accepts MycoFog. As long as it's a closed system, airtight, and with temperature control, that's our target. We have them in closed cell culture workstations as well. Here's a hypoxia workstation from the Baker company. For an example, this is your main working chamber. This is an interlock. You'd put the MycoFog through the interlock, bring it into the main chamber, and it looks like that just before you activate it. And these are glove ports, which are gas tight. And so even though they're extended and somebody's got their hands in them, this whole area is gas tight. So when you put the MycoFog on, the fog will not escape. Now, let's just look at a little bit of science and supporting data for you to give you that extra confidence that you need to know that we've checked everything out. So biological indicators, Randy mentioned them earlier. We have a very robust organism here called Geobacillus thermophilus. And I won't say that again. I'm going to use initials GS now to describe them. But that's a very robust bacteria. It comes from one of the suppliers in the US. And there are 10 to the 6 spores of this bacteria carefully placed on a little disc in the upper section of a biological indicator here. And what you want to see is that once you put this biological indicator into an incubator where you're performing a MycoFog decontamination, you want to see that this media, this purple color, remains purple. Why? Because you want the H2O2 to kill all the bacteria on the spores. When you activate this by crushing, the disc will drop into the media. We want to make sure that all the bacteria are dead, so the media should not change color. So these three show successful kill, and because there are 10 to the 6 spores on that disc, we call it a 6-log kill. And that's what you need to do to justify a biodecontamination process. It's recognized that that is the way of standardizing how well your protocol does. Now, if you have a failure in the biological indicator, or you have the biological indicator outside the target instrument, so that would be your control, then of course no hydrogen peroxide reached the bacillus, and so they're still alive. When you crush this top bit, they fall into the bottom there. You can see the disc in the media, and the bacteria grow, and you get the media changing to yellow. So yellow means bacterial growth, no kill, and purple is successful. Okay, and there's a death curve for the 10 to the 6. That's the time it takes to kill those 10 to the 6 spores. We've got a whole bunch of data on different incubators and whether the biological indicator was successful or not. N means successful, Y means there was no killing. So it would be yellow. And this table is there for you to see. I'm just to show you that the GS was placed in different parts of an incubator. So upper right front, for example, just to show you where these were all placed. Okay, and then this list grows as we get more data back from the field. Another very important validation issue is, well, is there any hydrogen peroxide left when I've finished the cycle? Will I get exposed to it? I meaning me, the user of a MycoFog decontamination system. And the answer is we put a hydrogen peroxide meter inside the incubator alongside the MycoFog, so we could track the parts per million of the airborne hydrogen peroxide over the cycle. In this case, we're using the 45C 3-hour cycle. And you'll see that about after 155, 160 minutes, there is no parts per million of hydrogen peroxide in the atmosphere. And that's very important. And we say, wait till 180 minutes is gone. So we've given another 10, 15, or 20 minutes even for complete dissipation to occur, and therefore no exposure to the user. Exactly the same data for a 37 4-hour protocol. Here are the parts per million. Here is time. This time in 4 hours, we've cleaned it out within something like 215, 20 minutes. Again, we give the extra 20, 25 minutes to be absolutely sure that there's no residue when you open the door to retrieve the MycoFog. So very important, we kill biologically and we don't hurt the user at all. So very quickly, and summarize this, and one more slide after this, I hope you can see that MycoFog is a very easy to use system. There's no reagent prep. It's walk away once you've pressed that button and closed the door. It's very simple to use. It's relatively fast compared to other biodecon systems and certainly biodecon cycles on incubators. It's affordable. You'll see that later when you discuss with Syntico what about the prices. And it's cordless, no trailing wires, so very easy and efficient. So that will allow us to say to you, please ask your customers, and please will you decontaminate more incubators more frequently, because there's now a method you can do that with. Lastly, here is the product offering that we were giving you, and you can get starter kits which combine instruments and reagents, and you can get reagents and instruments separately as well. Okay, we have different volumes of reagents here in the pods on the bottles because you need different volumes to deal with different incubator sizes. So we can decontaminate up to 500 liters with one MycoFog and one dose of reagents using our highest volume of 45 mil, for example. Could get into that discussion, which one to use, if you like, later on. All of this is going to be offered to you right now with a 10% discount offer, which everyone attending this will get a voucher from Syntica, offering you that price offer on all of these products. So thank you very much. I'm going to hand it back to Martin. Thanks for your time.
Thank you, Samir, and thank you, Randy. We're going to jump right into our Q&A, and that was a really insightful and informative presentation. So thank you once again. We have 4 or 5 questions here, and I'd encourage, again, our attendees to pose any questions they may have. Question number 1 is, are there any residual antimicrobial activity after the cycle is complete?
Randy, do you want to take that?
Yes, I'm just getting my sound back together here. Can you hear me?
Yep.
Great.
Good.
Sorry, Martin, could you please repeat that?
Yeah, for sure. So the question is there any residual antimicrobial activity after the decontamination cycle is complete?
Great question, because of course safety is one of our biggest concerns. No, there is no residual material, so it's completely safe to open your incubator and replace your cell cultures right after. Hydrogen peroxide degrades spontaneously to water and oxygen, and we've designed our process so that there, at the end of that cycle, there is no residual material.
Okay, perfect. Samir, just before I continue, can I have you maybe either stop sharing your screen or feel free to share the presentation again?
Yes, I can.
Yeah, no worries. And then the next question is, how often should one decontaminate an incubator? What is your recommendation? I'll let either of you guys tackle that.
Yeah, I'll take that. It's well, we recommend monthly or quarterly, depending on your laboratory workflow. Of course, we understand that you know, not all experiments fall into a 4-week cycle, and you may be completely unreasonable to think that one could do it monthly. But the concept is that a regular cleaning, so once every 2 months, once a quarter, or once a month, will prevent contamination so that you don't have to throw away cultures, waste valuable reagents.
Okay, perfect. Thank you. And Randy, you may have mentioned this earlier, but is the MycoFog effective against mold spores specifically?
Yes, and that's a really good question. Mold spores are resistant to killing by alcohol, either IPA or ethanol, and that is also the most common biodecontamination method. And so, interestingly enough, as we launched MycoFog earlier this year, we found instead of what we thought was the most common problem, mycoplasma, the most common issue is mold and fungus, and the hydrogen peroxide and the MycoFog system work very well against that.
Perfect. This is not a question per se, but one of our attendees, we asked what method they currently use for decontamination, and the answer was that they use an autoclave for the shelving. That's removable, which I guess is great. So that kind of deals with the shelving decontamination, but not necessarily the rest of the device, right?
Correct.
System.
Yeah, that's a good point. That's correct. It does just deal with that. And when you look at an incubator, and I've just done an incubator install, you've got to take the side panel. If you want to do a proper autoclave, you need to take the side panels into account. There's a cover on the humidifier down at the bottom. There's a bottom tray. sometimes a top tray, and other items that need also to be treated. All these stainless steel items have to come out and be treated the same way. If you just do the shells, you're not doing justice to a full clean.
Perfect. Samir, you did show that chart towards the end there with the different product configurations and volumes, but so are there limitations to the size of the incubator or workstation that one can use this MycoFog?
Then I think at the moment we're saying with one MycoFog, we're going up to 500, a chamber of an internal usable volume of 500 liters is our highest target at the moment. We're working on higher volumes, but right now with one single MycoFog and a bottle of reagent, you can do up to a 500 liter incubator. There's a smaller reagent size for up to 200 liter. There's a mid-size volume, 25 mil, for the incubators between 2 and 400, and then that higher volume of 45 mil for 400 to 500 liters. So 500 liters is the uppermost at the moment.
Perfect. Okay. I have a question, and maybe this is my naivety here, but you mentioned two protocols, 137 degree, 142 degree C, one shorter than the other, but I mean, any reason to choose one protocol over another other than the time that it would take?
Randy, do you want to just check that out?
Yeah, yeah. Excuse me. Sure. So what we developed our process at 45C initially to arrive at a reasonable time that it would take to disinfect and degrade the hydrogen peroxide. So those are the two steps that are going on in the chamber. And we found that we could settle on a 3-hour protocol at 45C. Then people started asking, you know, it's kind of a hassle for us to change our incubator temperature. Do you think we could just keep it at 37? And so we devised a protocol that is just as effective and just as safe. It takes an hour longer. And those are the reasons for the two different types of protocol. It's about the workflow in your own laboratory and your own preferences that we're trying to accommodate.
Perfect. Great answer. Thank you very much.
Right.
Okay, I'm just looking here. There are no other questions. I did say that the webinar would be approximately 30 minutes. We're 39 minutes in. So with that, I'll just thank everyone for their time today. Randy and Samir, thank you so much for the presentation and for sharing this information with the audience. It's a very important topic. We're here at Syntica to answer any questions. Randy and Samir are as well. If there are other questions that you have after the event, feel free to send them our way. And as I mentioned earlier, we will transcribe the Q&A and provide a link to the recording for everyone in attendance afterwards. So thank you again for taking the time. Samir, Randy, thank you. I don't know if there's anything else you want to add.
No.
Very nice.
Thank you so much.
Well done. Thank you, everyone.
Okay,
Have a good day, everybody. Bye-bye.